Okay, so these are the slides for our second tech talk, and so the focus for this tech talk is going to be fluorescent recovery after photo bleaching, or FRAP. So just as a reminder, we have our three different approaches for studying proteins. So this falls under microscopy-based approaches. So it's going to be using fluorescent dyes, either because of antibodies that are tagging to a protein, and those antibodies would have a dye on them, or because we have fused a protein with its gene with a fluorescent protein like GFP. Okay, so we'll talk a little bit more about that because I have the last couple slides are figures from a paper that's using FRAP technique to look at protein dynamics within membranes. Okay. So you might remember from our chapter 11 talk, I showed this figure where we have these proteins that are localized in the plasma membrane. We want to keep in mind that distributions of proteins within a cell can be different. But by using the technique of photobleaching, we can bleach an area of the cell, removing basically deactivating those fluorescent proteins because we've damaged them with the laser with this light. So they're now inactive, and so this allows us to look at dynamics as far as proteins moving around in the plasma membrane, so how that recovery happens as these damaged fluorescent proteins move out of the area because they are tagged with a protein of interest, and active fluorescent protein tagged to the protein of interest moves into that location. Okay, and again, we could photo bleach a specific area. So if we were interested in protein A here, we could photo bleach the apical edge here, and it would allow us to look at the recovery of protein A, where if we know the protein we're interested in is protein B and it's found in the basal plasma membrane, we could photo bleach that area and look at recovery for that.
The lateral mobility of membrane
So I'll put that video up.
So
this video shows the technique of FRAP and how it's done, so that video will be posted up under the files for Unit 2. But what we saw in that, what you'll see in that video is that the laser is focused in an area, and so it ends up kind of killing off, deactivating those fluorescent proteins in that area, and then depending upon the protein, if it's one that can move freely around in the plasma membrane, we would then get redistribution, and so the light intensity would increase because we have this replenishment of active fluorescent proteins. If it is one that's kind of anchored or in an association with other proteins, that we're going to see a different distribution pattern. So ones that are like in a lipid raft would not have as much movement as ones that dynamically can move throughout the whole entire plasma membrane. Ones that are anchored because they're anchored to like cytoskeleton are going to be much more localized. Okay? And depending upon what type of membrane they're found on can also affect recovery time and the dynamic movement of the protein. So through FRAP, we can learn a lot about proteins and where they're located, how they move, and how the recovery occurs. Okay, so as you saw in that video and as I was just mentioning, this figure just shows you how FRAP is conducted. So as you can see on the top here, we have the fluorescently labeled membrane proteins, and they can be fluorescently labeled because we have antibodies with fluorescent dyes on them that are recognizing the protein of interest, or we could have a fusion protein. So that fusion protein would have the gene of interest and then the gene for the fluorescent protein, like GFP, fused together. So whenever our protein of interest, when that gene gets turned on and expressed, you also get expression of the GFP, and they actually become this fused protein. So they're connected together. So when we have that, and again, traditionally, FRAP is looked at for the plasma membrane. So we'd have these proteins of interest with the fluorescent labels in like the plasma membrane. We would then take our laser beam and focus it in on an area. So we would end up damaging, bleaching those fluorescently labeled proteins. And so we would end up with this, what we have is like a clear window here. So you can see how there's no dye inside that area. And after time, depending upon the protein, maybe the protein can dynamically move through the plasma membrane. And so you'd get some recovery of that in that bleached area, because undamaged fluorescent proteins would come in to the area and some of the damaged ones would leave that area. And so what we're seeing here in this third cell down is in that window we start to have some, it's not the same intensity, but we have some recovery occurring. Eventually you would get complete recovery because there would be the replacement of those damaged proteins with new proteins, right? So the proteins in our body are constantly renewing themselves, so taking damaged ones out, putting newly synthesized folded proteins in. So we would eventually get full recovery depending upon the protein, how stable it is, how often new protein is made, how much of it is made. That would affect recovery time, or at least full recovery time. So when we look at the proteins, we can actually look at individual proteins and study how they're how they're trafficking. So in panel A, if we trace that movement of a labeled protein, we can actually see that this one is very dynamic. It can move across the plasma membrane relatively easily. So that would tell us about the trafficking pattern for that protein. Opposed to our panel B and C, where we don't see as much movement present. So in panel B, this would be an example of a protein that's going to be more isolated with interactions with other proteins or other aspects of the plasma membrane. And so this might be some protein that's being found on like a lipid raft, right? So it's not going to have free movement throughout the whole entire plasma membrane. It's going to be isolated in a smaller area. Now with panel C, what we're seeing is one that would be anchored most likely to the cytoskeleton. And so it's not going to be able to move freely at all. It's actually very limited. The most severely limited one. And so again, by looking at trafficking pattern, this can also tell us information about the protein. So our next few slides just are an example of how FRAP is being used. So this paper is from 2018, so relatively new. Full disclosure, I picked this paper because it looked at KDL. So we're going to be talking about KDL in chapter 15 when we're talking about protein sorting and how do proteins get to where they need to. And so the KDL is talking about a 4 amino acid sequence on the C terminus of a protein. And this tells it to go to the ER and stay in the ER. And so if you're like a protein that's going to be helping with folding of other proteins, like a molecular chaperone, you would have a KDL on the end of your amino acid sequence that would let other proteins know that that's where you're destined, that's where you should go. So proteins helping you to get to your location would know to bring you to the ER and leave you there. Okay, so the protein I studied in grad school, GRP78, has a KDL, and they actually mention that protein in this paper. And so again, it's one of the reasons the paper kind of got my eye. The other reason I picked this paper is because this paper is looking at the use of FRAP for membranes that are not the plasma membrane. So again, traditionally FRAP is looked at on plasma proteins in the plasma membrane, but it can also be used in other membranes like the ER. And they also look at the nucleus, so nuclear membranes, okay, or nuclear envelope. So in our top row, when we see the red, what we're seeing is a probe that's going to recognize that has M cherry as its fluorescent protein, its fluorescent dye, and it's going to recognize the KDL. Okay, so that's going to be localized in the ER's lumen. So again, if we think about the ER, there is going to be a lumen and inside, but there's also going to be the membrane. Okay, the second row is going to be where we're going to see the actual ER membrane, and this is because of a probe emerald, emerald for the green, and SEC61 beta. SEC61 is a transporter, a uniport, so it is going to create this kind of channel that is going to allow for newly synthesized polypeptides to enter into the ER. So think about protein synthesis. We're going to have our ribosome moving down the message RNA, and it's adding on these amino acids, creating a peptide bond between it. And once that polypeptide chain gets long enough, we're going to have the ribosome moving and docking onto the ER. This is where we have a rough ER, and that polypeptide chain is actually going to start to enter into the ER, so that folding and any kind of modifications that need to happen, it can move through that process. And again, we're going to be talking about that process in chapter 15 about how proteins get to where they need to be, but SEC61 is really important in that, and it's embedded, it's a transmembrane, it's embedded into the ER membrane. So by probing for that protein, it allows us to look at the ER membrane. By probing for KDL, that lets us look at the lumen. Okay, so proteins that are going to be found in the lumen, in that space of the ER. And then lastly, that last row is a merge. So what they do is they take those two images and stack them together. And if you add red and green together, you get yellow. So where we have like a co-localization gives us this yellow appearance. So we do want to keep in mind that these fluorescent dyes and these proteins have some mass. And also depending upon the quality of the microscope, you know, how small or plain of view, it may not actually be like co-localizing in the same locations, in the same general location, if that makes sense. And I'll touch upon a little bit more about that in a moment. Okay, so looking at this, we can see at 0 seconds, it's kind of like a control. This is what it looks like without any kind of photo bleaching occurring. They then are going to photo bleach. They're going to FRAP that area where the circle is. And what you can see at 6 seconds is that in both the case of the M cherry and the M emerald, we see a decrease in the amount of fluorescence that is there. So, you know, showing the FRAP technique is working. Now when we then move to 12 seconds after the bleaching, we can see that with the M cherry, with the KDL and the lumen, we are seeing recovery. We're seeing that there is more red compared to 6 seconds in that that circled area. Now, when we look at the membrane, we don't see that recovery occurring. And so if we move down to the merge, we can see in that window, we can see more red. So the merge is helpful when we're looking at this recovery, because the recovery is not happening at the same time. And so we can see that we get recovery of our KDL, our ER lumen sooner at the 12-second mark versus the ER membrane, the emerald. It's taking 50 seconds for recovery to happen. So, I mean, so when we think about 12 seconds, 50 seconds, I mean, those are relatively short times, but the recovery is different depending upon where the protein is found, right? So in the ER, we're going to have or in the lumen, we're going to have a fluid movement of the material, where in the membrane, it's going to be has to move within the membrane to recover, right? And it's that movement of undamaged proteins in and damaged proteins out. So we're getting a mixing of the kind of the bleached material and the unbleached material. Okay. So the next thing that they show in this paper is they look at the ER membranes, which in the previous figure they show that they can look at ER membranes using FRAP, and they also look at the nuclear envelope, which would be the nucleus, and they look at its recovery. And so if we look at pre-bleached and then bleached, and then for post-bleach they select 27.3 seconds. I had mentioned, I'm like, I don't know why that's kind of weird time why they didn't pick 30 seconds. I imagine someone just pushed the button a little too early and they just wanted to make sure that it was accurate. Maybe something magical happens at 30 seconds and they wanted to get up before that. And then we have a 360 second post-bleaching. Okay, so a short time and then a longer time. And so when we look at the ER, we can see that window, that square that's bleached, we start to get some recovery at the 27-second mark, but by the time we have the 360 second mark, it appears to be like a full recovery. Okay, now comparing that to the nuclear envelope, we don't see that recovery occurring even at that longer time point. So this would show that the proteins in the nuclear envelope, they do not move as easily, so they're more anchored in position. If we think about the membranes of the nucleus, there you're going to have all those nuclear pores in them, and so it's not going to be the same dynamic as we would see in the ER. Okay, and so this means that there would be a longer recovery time. So if maybe they looked at 10, 20 minutes later, then at that point they might see more recovery. Okay, but what they do show is that you can do photo bleaching to specific membranes within inside the cell, to the ER membrane, to the nuclear membranes, the nuclear envelope of the cell, in addition to the plasma membrane. So one of the last little pieces I just want to talk about is that the equipment that's needed for the FRAP, usually a confocal microscope is going to be used. That confocal microscope is going to be hooked up to a computer system with software that does a lot of this imaging for you. So it's not, yes, you look through the microscope to set up your focal plane and get things into focus, but then a lot of the manipulation is happening through a computer. So you're not directly even touching the microscope, because the microscopes are so sensitive that they're actually on float tables to minimize any kind of vibrations, because the resolution, the area they're looking at is so small that even slight vibrations, like a truck going by, could cause that microscope to move enough to cause the image to be blurred. And so these setups are not cheap. A cheap entry-level confocal microscope is going to be over $100,000. A decent confocal microscope setup is going to be closer to $500,000, and really nice setups are going to be in the $1 million mark. Okay, so another factor besides just like lens quality and maybe your vibration table and the computer setup that would cause an increased cost is the number of lasers. So confocal microscopes could have as few as 2 lasers. Generally, they have at least 4 lasers nowadays, and that gets you up in price. So each time you add a laser, some of the systems, the really nice Cadillacs of the systems, they're going to have 8 or more lasers to them. Each laser lets you look at a different color. So for our paper here, we would have had to have a laser that allows us to look at M cherry and a laser that allows us to look at M emerald. But you can go to a database. In the Tech Talk quiz, there's a link to this database of different fluorescent proteins, and there's over 700 different fluorescent proteins. So there's tangerine. There's all these different ones named after fruit because of the color of the fruit. There is not an M lemon for some reason. Maybe that's in the works, but there's a papaya. I kept going through different fruits. And so there's a lot of them. There's not watermelon either, which is kind of weird, but and there's your good old-fashioned green fluorescent protein, red fluorescent protein. So there's all these different fluorescent proteins, and a lot of them overlap in the wavelength that they get excited at and then emit. So you don't need 700 lasers, but 4 lasers starts to limit the number of fluorescent proteins you can look at or dyes you can use in your experiments, where if you had 8, you could it's not just that you can look at 4 additional ones. It's an exponential factor that you can, the number of dyes that you could use. So you could, if you have 8 lasers, you could have a system that you have 8 different proteins you're looking at and how they're interacting, and each one has a different dye tagged to them. So you would be able to see those interactions and how those occur. So again, that's why you can have a price range difference in these scopes. So Kennesaw State actually has a confocal microscope as part of their microscopy core facilities. Their scope has 4 dyes to it. So in theory, you would be able to do a FRAP experiment. So again, with a FRAP experiment, you're just leaving that laser on in an area for longer than you normally would, which bleaches out. So just normally when you're using a confocal microscope or a fluorescent microscope, if you leave the light on in an area, you end up bleaching out. A general use, a non-confocal fluorescent microscope, doesn't have the range to pinpoint. So that's why a confocal microscope is traditionally used, because with a confocal microscope you can you have more control of where the laser is focused on. So that allows you to bleach a specific area. Okay. So hopefully you have found this Tech Talk interesting. You've learned about FRAP, you've learned about fluorescent dyes, and how this technique can be used to look at protein dynamics in membranes. But I would also point out, we can also look at these protein dynamics within space of the cell, so within those lumens, and also in the cytosol.